Hello and welcome to Nikolay's genetics lessons, and today I have prepared 3 questions for you. And as usual, I recommend you to stop video here, read the questions, choose the correct answer, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So here is the first question. A cell can change the expression of its genes in response to external signals, and you have to choose between true and false. And of course, the correct answer would be A, true. Otherwise, life wouldn't be possible if the cell wouldn't be able to respond to external signals. And next question or statement: different cell types that respond to the same hormone usually turn on the same set of genes. Now I think it would be a little bit hard to choose the correct answer, but the correct answer, no matter what you choose, is B. This is false statement. Imagine that, for example, I wouldn't provide you example from the cellular biology, but just normal daily life example. Imagine that robber break into the house, and do you expect that everyone in the house would behave the same way and respond to this the same way? For example, children, their mother and their father, probably not. So children probably would hide, mother would probably call the police, and the father would probably try to defend the family, whether blocking the door or actively, if he has a gun, for example. And police, when they would come, they would actively defend and act in such situation. So as you see, cells also respond to the same hormone differently depending on their functions. If cell lines is differentiated and has a different function, of course the response to the same signal can be different. And next question: although all of the steps involved in expressing a gene can in principle be regulated, for the most genes the most important point of control is, and here is the 5 answers to choose from. Answer A: transcription initiation, and imagine that this is double-stranded DNA, and here message RNA is produced, and here we would have 5' end. Message RNA always grows from 5' end to 3' end direction. And answer B: RNA processing. So when message RNA would be made, it consists of exons and introns in eukaryotes. In prokaryotes there is no introns, just exons, so it would be processed and during the process of maturation, and introns would be exist here, and mature message RNA would be made as next stage. So answer C: RNA transport and localization. In prokaryotes there is no transport of the message RNA, it would be translated even during the transcription. So this loose end, even though it's still processed, it still grow, but this loose end could be a site of attachment for ribosomes, and it would be translated right away. But in eukaryotes, message RNA have to leave nucleus and would be processed, and protein would be made outside of the nucleus. Answer D: message RNA degradation. Of course, message RNA has its own life expectancy, and it is protected from both ends. From 5' end, this is going to be 5' cap, and from the 3' end, this is going to be poly A tail. And the longer this tail, the longer life expectancy of the message RNA. So this also can be a point of regulation, And the last answer E, message RNA translation. And this is going to be the last stage when message RNA would be used in order to process it. So this would be a point of, for example, ribosome attachment, and protein sequence would be made from the sequence of the message RNA, and this is also can be a point of regulation. So we basically have to choose between this first stage, second stage, third, fourth, and fifth stage, what would be preferential for the cell mechanism to regulate message RNA life cycle. And of course, as you see, the easiest way to regulate gene expression would be the first stage. Although gene expression can be regulated at all the stages, but message RNA production, processing, transport would take energy and would take resources of the cell, and of course it would be much easier to control gene expression at this first stage. So our answer would be answer A, transcription initiation. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.